Okay, so this is the second piece of the hormone lecture. We've already talked about the two different types of hormones. You have lipid-soluble hormones. Those would be the steroids, such as testosterone and estrogen. Then you have water-soluble hormones that are polar, and those are most of the hormones, including things like oxytocin, we talked about, calcitonin, things like that. Okay, so most of the hormones are going to be water soluble. So now we need to talk about how their polarity affects how they're able to interact with the target cell. Before we do that, let's talk about, well, what do I mean by a target cell? Remember that hormones, as endocrine glands, the hormones are going to be secreted into the bloodstream, and from there they have to just travel around into the bloodstream. So the question is, well, how do they know which cell they're supposed to go to? What is their target cell? And the answer for that is the target cell is going to have a receptor. And what I mean by a receptor is a specific protein that's embedded in the cell membrane of the target cell. And that receptor protein is going to have a binding site that it physically matches, corresponds to the hormone shape, right? Just like the lock and key model that we already saw with enzymes. And of a specific shape, it'll fit right in there, and that'll initiate how the hormone works. Okay, so the target cells have a receptor that is specific to the hormone, only binds that particular shape of the hormone. So the location of that receptor cell, though, depends on whether or not it's water soluble or lipid soluble. If it is water soluble, that receptor is going to be found embedded in the membrane, whereas the lipid soluble ones are going to be inside. Okay, so let's just quickly review the structure of the phospholipid bilayer to help us remember how that will interact with polarity. So here here's one phospholipid. There is the hydrophilic head. These are the hydrophobic tails, and when they're created, it's oriented so that the hydrophilic tails will all point inside, so that you have the polar heads on the outside and the non-polar hydrophobic tails pointing in the inside. And this creates a nice, thick hydrophobic core. So what types of molecules can pass through that hydrophobic core? Like dissolves like, so only the non-polar or lipid-soluble hormones can pass through. Because the water-soluble hormones are hydrophilic, they are polar, that means that they cannot diffuse through the phospholipid bilayer. So that means that they have to bind to the receptor that is on the surface of the cell, and then they need to use some other second messenger in order to create a response in the cell. So they're bind to the receptor that activates a G protein. The G protein then will activate a second messenger. Remember, the second messenger is the thing in the cell that does the response for that cell. Make some sort of physical change of that target cell. Okay, so there's multiple different second messengers out there. I don't want to go through all of them. I just want to go through cyclic AMP because that is the most common one for the water-soluble hormones. So we're just going to go through cyclic AMP. So this is a little bit of a complicated process, so make sure that you really think of it like a story. It's really about cause and effect. Okay, so don't memorize individual pieces. You want to understand how one thing causes another thing causes another thing. Okay, so the very first thing is, here is the hormone. Sometimes the hormone is referred to as the first messenger. So it is the thing that's released into the bloodstream, and then it's going to bind to the receptor. And remember, the receptor for the water-soluble hormones is going to be embedded into the phospholipid bilayer. Once the hormone binds to the receptor, that's going to change the shape of the receptor, and that's going to then activate a G protein. So here is the G protein in blue right here. When the G protein is activated, it has this GTP molecule attached to it. That's probably where the name G protein comes from, although I'm not actually 100% sure of that. Okay, so here is an activated G protein. I know it is activated because it has that GTP molecule bound onto it. Now that that G protein is going to be inside of the cell, and it's going to physically move around. And what it's going to do is it's going to go to an enzyme. In this case, for this pathway, the enzyme is going to be called adenylate cyclase. Adenylate cyclase, ASE, that's the enzyme. And then once that enzyme is activated, it's going to then take ATP, which is what you've already seen, the energy molecule. It's going to take off two of those phosphorus groups, the phosphate groups, two of them off, and then it turns it into a circular structure, and that makes cyclic AMP, CAMP. Okay, so cyclic AMP. Now the cyclic AMP, that is the second messenger, and the reason it's the second messenger is because that's what's going to cause a physical change inside the cell. It's going to activate some enzymes, or sometimes it causes secretion of hormone or secretion of a product or something like that. It's a trigger a response. So the second messenger is going to be the cyclic AMP. Okay, so here's the sort of very generalized version of what I just said. Okay, so it's really like one thing activates another thing, right? So the first thing you have is the hormone binding to and activating the receptor. The receptor then binds to and activates the G protein. The G protein will then be bound to a GTP. It'll then travel over to the enzyme. In this case, the enzyme is going to be adenylate cyclase, and the adenylate cyclase is going to convert ATP into cyclic AMP, and then the cyclic AMP, that is the second messenger because that causes the response, which is mediated by a bunch of kinases, which are just enzymes that add phosphate groups to different things, and then that will be your response. Okay, so why do water-soluble hormones need a second messenger? They need that second messenger because they are unable to get into the cell to cause the response themselves. Instead, they need some other thing to trigger the response, and that's the second messenger. Okay, so don't lose sight of that important fact. Okay, so there's some videos that you can do. You can just go into the PowerPoint and play them directly through there. You can also copy and paste the URLs. Okay, so now let's go into the lipid-soluble hormones. Remember, lipid-soluble hormones is also another way to say hydrophobic, or you can also call them non-polar. Non-polar, hydrophobic, lipid-soluble, all kind of the same area. So because they are lipid-soluble, that means that they are able to dissolve and interact and pass through that hydrophobic core. And because they can pass through, that allows them to diffuse into the cell. And once they're into the cell, they're going to then bind to the receptor. So the receptor for lipid-soluble hormones is going to be inside of the cell. Once the receptor is bound to that, once they are bound together, they create a little complex, a receptor hormone complex. And then they travel to the nucleus. And what do you think they do in the nucleus? What's in there? DNA is in there. And what do you use DNA for? You use the DNA to do protein synthesis. So that's what they do. Those hormones, the complex will go into the nucleus. It's then going to activate protein synthesis. Okay, so first you do your transcription, then that makes that mRNA, and the mRNA is then going to go to the ribosomes, and then that's going to be translated into a protein. Okay, so because the lipid-soluble hormones are non-polar, they're allowed to diffuse right in and have an effect on the cell themselves. They don't require a second messenger. Okay, so that's enough for this little piece. Next, I'll talk about the message. What's going to happen inside?